I will start with actually introducing phenotypic screening, which encompasses the high content screening, and then talk about the data, how we acquire it, how it looks like, what is the point actually. And then I will talk about challenges of this kind of data and application of deep learning in this research. So starting with drug discovery, because the main point is to find drugs and find drugs that work and are not killing people. We have two main streams. One is target-based approach and the other is phenotypic-based approach. With target-based approach, we are working mostly with molecules. We are optimizing them. They are lower. This is lower cost approach. However, we need to know the target that we want to fix in the human. And it's hypothesis driven, and it has lower possibility for actual clinical translatability. With phenotypic based approach, we do not have target, we do not have the exact thing that we want to fix in the cell or in the DNA and so on. We kind of know what the fixed human looks like or fixed cell looks like, and we want to mimic that. This is kind of much higher cost because we are doing this en masse and we are producing a lot of data and trying to compare a lot of reference points to our healthy human, but it has a higher possibility for actually leading up to the clinical approaches and to drugs. So how we are creating data with the phenotypic approach? We have something that's called high content screening. The screening part comes from the fact that we are screening molecules for what we are looking for. High content comes from the amount of data that is produced and the automation that leads to that. So how we are producing that data? First of all, we have our cells of interest. This also can be bacteria or smaller organism, but I will be focusing on cells. And we are putting them in something that's called a plate. Plate has a lot of smaller wells that are very, very small, and we can put cells into them. And then we expose the cells to something. And this something can be chemicals, for example, drugs. This is our main focus. Some environmental samples, nanoparticles. It can be also genetic perturbations. So think something like the COVID vaccines that are targeting mRNA. After that, there is some incubation. The cells have to grow, have to interact. There has to be time to actually, for the compound, the chemical compound to do something. And then we are staining the cells. Here we have cell painting, which I will be introducing in a second. But the main point of staining cells is that we want to dye different compartments of the cell so we can see different parts. Of course, we can take a regular picture with microscope that's called bright field, but this will result in like a grayscale image where we cannot really see a lot of details. With dyeing the cells, we actually can see a little bit more, even like the parts of DNA and so on. And those cells are stained with fluorescent dyes that do not do anything special to the cell other than attaching to one part of the cell and showing it. And then comes the high content part. Everything is automated. There are automatic pipettes, automatic robotic arms that move everything. Some of that, of course, is done by humans and still in smaller laboratories, but the main point is to do it automatically at very high speeds. And then we get those pretty pictures that are here that are showing the cells. There's a lot of them, and then we go into the step of the actual image analysis. This can be done with some human-defined features, and the cell profiler is one of the examples of that approach where we measure things that are that we can see and that a human can define, like shape, size, texture, and so on. Or we can, of course, do deep learning, which I will be focusing on. And from that, we get a profile of the cell, we get description of the image, which can be analyzed furthermore. And now 2 examples of how the data that's coming out from the high content screen can look like. So we can have a simple experiment where we have only 2 dyes. One of them is showing the nuclei, so the center of the cell. The other is showing plasma membrane, so the let's say body of the cell. And here we have also an example of the change that we actually can describe. So the top one, the control, are cells that were not touched. There was nothing done to them, and as we can see, they are kind of long. Those are microglia cells, which are part of the nervous system. So we can see that they are kind of elongated. The nuclei is somewhere in the middle and so on. And then we have cells that are inflamed. This kind of inflammation can lead to things like neurodegenerative diseases, for example. And what we can see on the cells is that they are rounder, there are some spikes, and those are actually things that we can describe. Then the more complex example is cell painting, where we have 6 different stains. The image that is a result has 5 different channels, so we cannot treat those images as RGB images like with natural image problems. This cell painting shows 8 different parts of the cell. So we are painting virtually the entire cell, and we can see each part of it. The point of cell painting is to actually create experiments that are more universal, that are not focused on one problem, that can be used for different tasks, for different problems, that can at once analyze a lot of different things, not only if it's working, but also does it kill the cell or not. And now let's go to the challenges of this data. As I said, high content. So there is a lot of this data, a lot is produced, and right now we actually can say that there is also a lot of public data. Here are examples of data sets that were created by recursion. And I think 2 or 3 out of them are actually public. The RX3, I don't think so, or small parts of it are public. However, at the last row here, you have the size of the data. And this one plate of data that I was showing at the beginning, it can produce around 20 to 25 gigabytes of data itself. And here we have even 83 terabytes. So first of all, we have very, very large amount of data, and this is images. These are not labels for this data. We only know what the compound is and how it looks like on the image. Here we still don't know what it's doing. The second problem is that each lab is doing their own thing. Even though a lot of them are using cell painting, and statistically it's the most used type of staining for deep learning research, every lab is doing their own thing. There are small differences, so it's maybe someone gave a little bit more of the dye. Maybe someone, opened the window, there was more light coming and so on. So we get a big batch effects and, out of distribution problems where we cannot apply models trained in one lab to the other. However, here we have example of the data that was created in last 2 years by the JUMP Cell Painting Consortium. And they created a very big effort. They pulled 10 pharmaceutical companies and 2 nonprofits, and companies shared the compounds between themselves. So each compound was screened at 5 different laboratories. There was a lot of data, 130 terabytes, and they shared this publicly. So this is like one of the first steps to actually creating something like ImageNet, but for high content screening, where we actually can do something that is not focused on one lab. However, this is public data, and there are problems with that. For example, the companies that were sharing the data shared the stuff that they wanted to share. So not very interesting, and with very low number of labels. And here we have example of the public data set. It only has 30,000 of compounds and its labels. So the blue ones are examples of labels for compound that is active for something. And orange one are the compound is inactive for something. And as you can see, we have a lot of tasks that do not have any labels. We have a lot of negatives in comparison to the positives. So for deep learning model training, this is a nightmare to actually, derive some statistics, create cross-validation. It's very hard to divide those compounds, and training those models is not as easy as we wanted it to be. And then the fourth problem is interpretability. Biology is complex. We cannot describe everything. The image that I showed you with 2 dice, that was easy to interpret. The further we go and the bigger problems we are solving, it's harder to interpret. As I was saying, Cell Profiler has some features, creates some features that are interpretable. We can say that, well, this set of cells is bigger than this one, but does it show all the examples? And if we show the images to biologists, they still say, oh, well, maybe this is that, but I'm not sure, we cannot test it, and so on. And the last thing is complexity of data and of targets. The drug can be working very well, but it can have negative side effects and kill cells, or even, we take 2 drugs that are interacting, they will be killing cells or killing us. So what are the applications? After I talked about the problems, let's say what we actually can do with those images. First of all, we can identify hits, so we can start at the very beginning and find compounds that are actually doing something. And this can be done either by, first of all, training on the negative controls. Negative controls is, we didn't do anything to the cell. It's its basic state, and we can train models to learn how this neutral state looks like, and then it picks up anything that is different from the neutral state. However, here, the big problem is that dead cells, example of the image here, are also very different from negative controls. So with this application, we need to look for hits that are not actually toxic. The second hit identification case is where we actually know what we are looking for, kind of. So we have a drug that works, and we want to mimic that, so we can train those similar models to compare images to negative control and to our positive control, so something that we are looking for. And the next one is property prediction. So we can, from the images, we can actually predict properties of the compounds. And this can be something as simple, let's say, as solubility, but it can also be something more complex. What is it doing to the DNA? Is it killing it? How it's influencing ATP production, and so on and so on. So we can have this panel of properties and train our classifiers to predict it from images. However, the end-to-end training also has its problems with, in this case, that we are very much limited to the task that we were training on, and then we cannot transfer that model anywhere else. So we can split it into 2 parts of the representation model training, and then creating a representation that is describing the cell as well as it can, and then classifying that. However, the training representation model in this case is rather challenging, because, well, if we cannot ourselves say everything about the cell, we can even measure everything when we are evaluating those models. So they need to be evaluated on their performance on the tasks. And examples of how this representation can be trained are basically whatever was in the research for natural images. So we can have CNNs like ResNet trained for different tasks, and then we test if they are working, if that representation is working well enough. We can also go into contrastive approaches like SimClear, and with JumpCP dataset, it's easy enough because we have enough examples to actually pull those positive examples for contrastive learning. And of course, as with all the images, there was push for transformers. We have here distillation with no labels that uses vision transformers, and right now there's also research with masked transformers and so on and so on. And the important here thing is that dot below. And this is the CNN model that was trained to predict structure. And what we can see here is that structure is not showing everything, and boosting it with images actually helps. And the image can actually see more than the structure itself. With the next application, we combine images and structures. So we can train a model to compare image and structure, are they similar, and then use this model to generate new compounds, well, generate or find new structures that could induce that phenotype. And lastly, there are some approaches to predicting image straight up from the structure. And here are examples of how they look, and the ground truth. This is very subjective if it's working or not, but there are also those applications as well as things like trying to predict some stains like cell painting from the straight up bright field image with the grayscale to the colorful one. So these were the application where we can use deep learning for high content screening. And in summary, deep learning helps very much in this case. We are accelerating the research. It's much faster than the things like cell profiler that I mentioned earlier. And it reduces cost and time. We don't have to spend hours of compute time to generate a few thousand features. We can do it by passing it through neural network. There is a big problem with labels, with public labels, because, you know, big pharma companies have their own labels and they are producing much better models. And there's still a way to go for interpretability, and this is very hard because we still cannot create, for example, prototypes for cells. This is just too complicated biology. And that was the summary. I will, for a second, change the aesthetic of my presentation. As was mentioned in the introduction, I did my PhD at Jagiellonian University. I submitted my thesis a month ago, so I'm not a student anymore, but I wanted to invite you to the other talks of GMOM students and researchers. We will have 2 talks in the afternoon today, 3 talks in the afternoon, and 2 tomorrow. So please feel invited to those talks. And thank you very much for listening.